This is the review podcast. I would say that because something is or is not in this review doesn't necessarily mean it's definitely going to be on the final. But I understand that, especially in a summer class, it's really difficult to review all this material. Again, to give you the format of Test 5. Test 5 will be 50 questions, multiple choice, 6 questions, extended response. 25 of the 50 questions, multiple choice, will be on the Test 5 new material. So the podcast from Test 5 and the PowerPoints from Test 5, for week 5. And then 3 extended response also from week 5. The remaining 25 multiple choice questions and 3 extended response questions will be from the previous material in the course, tests 1 through 4. So I fully recommend that in studying for any test for any professor that you try to predict what he or she will ask. And that should be a reflection of what he or she thinks is important and what he or she has emphasized. In my case, I have given you the material in different formats. In this online thing, I gave you material in terms of it being in the textbook and it being on a PowerPoint and it being on a podcast. And then on top of that, for some material, I also gave you these handouts, which are under the handouts tab. And that is an indication of what I think is the most difficult to learn and also the most important in terms of carrying it into other classes. So I'm going to go over these podcasts now briefly. If you want more detail on these, you can return to the previous podcast where I went over them for the first time in more detail. So the first one, the central dogma handout. This is the idea that the central dogma idea is that genetic information is encoded in the DNA. That DNA is transcribed into RNA. That RNA is translated into protein. And protein is the thing that's going to do most of the work in cells. So if we look at a eukaryotic cell with a nucleus, inside the nucleus is that DNA, and inside the sequence of that DNA, most of that DNA sequence is not coding for protein. Most of it is non-coding, and therefore a gene can be complicated in that it can carry in it coding sections that tell the cell how much of the gene should be transcribed and when that gene should be transcribed. And those can be physically far from the part that turns into protein. In addition to, there's some genes that just don't turn into protein. But if we consider the genes that do turn into protein, if you zoom in on the DNA, 5' to 3' left to right is considered the sense strand. DNA is double-stranded. The antisense strand is 3' to 5' left to right, anti-parallel. So this is called the sense or non-template strand, and this is called the antisense or template strand. It's called the template strand because it is the template for making mRNA. So this strand of mRNA is transcribed into this strand of, this strand of antisense DNA is transcribed into this strand of mRNA. Remember, all the T's all the A's that would bind with a T on the opposite strand. There's no T's in RNA, so there are U's there instead. And in an mRNA, if it's a eukaryote, there are both introns and exons. Exons are the things that code For protein, introns are intervening sequences. Introns get cut out as that mRNA is processed and the exons get put together. Once that mRNA has been processed and the exons are put together, then that exon will leave the nucleus through a nuclear pore and be read by a ribosome out in the cytoplasm. That ribosome is going to look on this sequence for the start codon. The start codon will always be AUG, and that AUG codes for methionine. AUG binds with an anticodon on tRNA UAC that is antiparallel, and that tRNA has a methionine attached to it. That's the first codon, and then every codon after that will be 3 at a time. So this would be codon number 2, and this will be codon number 3. So the sequence before the first codon does not get translated into protein, and that does not count as a codon. Only the first codon and then those after those count as codons. Okay, now moving on to our next review sheet on mitosis. So mitosis is replication division. Whatever you start with, you end up with the same number of chromosomes per cell. So if you start with 6 chromosomes per cell, 1, 2, 3, 4, 5, 6, you'll end up with 2 cells and 6 chromosomes per cell. In mitosis, you start with chromosomes that have already been duplicated in the S phase, and that duplication is represented as 2 chromatids per chromosome, but same number of chromosomes. So before S phase, 6 chromosomes. After S phase, 6 chromosomes, just 2 chromatids per chromosome. Okay. In prophase, the nuclear envelope degrades. Metaphase, all the chromosomes line up in the middle of the cell. Anaphase, the chromosomes split down the middle, and temporarily, per cell, you have twice as many chromosomes as you started with, but that's only during anaphase. Immediately after anaphase, there's telophase and cytokinesis, and you end up with the same number of chromosomes per cell as you started with. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 2 cells, 6 chromosomes per cell in this example. Okay? And in meiosis, meiosis is reduction division. So whatever you start with, you end up with half as many chromosomes per cell. And if you start with 1 cell, you end up with 4 cells. So mitosis, you start with 1 cell, you end up with 2. Meiosis, you start with 1 cell, you end up with 4. So in meiosis, what is different about meiosis is crossing over. Crossing over happens in prophase 1, where you have homologous chromosomes. That means chromosome 1 from mom and chromosome 1 from dad line up on top of each other and they exchange information. So one arm of the A chromosome from mom is exchanged with that same arm A chromosome from dad. Those chromosomes are then separated during stay together during metaphase 1, separated during anaphase 1, and after telophase 1, where you go from 1 cell to 2, you've already gone from diploid, 6 chromosomes per cell in this example, to haploid, 3 chromosomes per cell in this example. Okay, that happens after meiosis 1. And then in meiosis 2, it looks just like mitosis. Prophase 1, the nuclear envelope disintegrates. Metaphase 1, they line up in the middle. Prophase 2, the nuclear envelope disintegrates. Metaphase 2, they line up in the middle. Anaphase 2, split down. Each chromosome with 2 chromatids splits down the middle, becomes 2 separate chromosomes. And you end up with 4 cells. Each has half the number of chromosomes that you started with. And those chromosomes are likely to look different than they did when they started because of crossing over. Okay, now if we move on to the stages of respiration, stage 1 through 5. Stage 1 glycolysis happens in the cytoplasm, where glucose is oxidized as you go down the page. It takes a couple ATPs to get the process started, and then you remake 4 ATPs down on the bottom of the page. After the molecule is split into 2 3-carbon molecules, you end up with 4 ATPs made, but a net gain of 2 because you use 2 at the top page here. You also charge up a electron carrier. NAD becomes NADH. NAD is reduced while glucose is oxidized. At the end of glycolysis, you end up with a 3-carbon molecule called pyruvate. That pyruvate gets oxidized and tagged onto coenzyme A. It gets decarboxylated, so carbon dioxide is removed. You breathe that carbon dioxide out as a gas, charge up another battery, and at the same time you're moving this 2-carbon CoA molecule across both membranes of the mitochondrion, the outer and the inner, and you end up in the matrix of the mitochondrion where stage 3 happens. Take that 2-carbon molecule, take it off the CoA, add it to a 4-carbon molecule to make a 6-carbon molecule, citrate, which is where the cycle gets its name. Citric acid cycle, citrate, citric acid, same thing. Also called the Krebs cycle, also called the TCA cycle. This happens in the matrix of the mitochondrion. What you get out of the Krebs cycle, or TCA cycle, or citric acid cycle, is a lot of reduced molecules. So 1, 2, 3 reduced NADs to become NADH, and 1 FADH2. These are high energy electron carriers. You get 3 NADH and 1 FADH2 per pyruvate, so that means you get twice as many per glucose. And you make 1 measly little ATP. That's if you're a plant. You make GTP, actually, if you're an animal. Okay? And stage 3 also creates CO2, which you breathe out. Now in stage 4, NADH and FADH2 release their high energy electrons to be passed down electron transport chain, okay, which is in the inner mitochondrial membrane, okay. And it's really important to know, and a lot of people mess this up on the last test, electrons go this way through the membrane, through the plane of the membrane, from the beginning to the end, and the end is going to be onto oxygen. That's why you need to breathe oxygen. So electrons are going down the page, up down the page this way, while protons are going across the page this way. So electrons and protons are not the same thing. Electrons are going this way. As they lose energy, that energy is used to move protons this way, which is from the matrix to the intermembrane space. That takes energy. The energy is received from those electrons that are losing energy. Protons are moved into where they're already high concentration in the intermembrane space. Now you've created a proton gradient, higher here, lower here. Those protons go through stage 5 to make ATP, and this is where, in stage 5, where you get most of the ATP. Okay? If you're going to break down something other than a carbohydrate, like you're going to break down lipid or you're going to break down protein, you're going to create a 2-carbon intermediate when you do that, and that 2-carbon intermediate enters the citric acid cycle right here, like you saw with glucose. Everything on this page, stage 2, 3, 4, 5, requires oxygen. If you have no oxygen, it backs all the way up to stage 1. Everything on this page does not require oxygen. Okay, so let's look now at photosynthesis. Photosynthesis looks a lot like, I'm going to download this, looks a lot like respiration. They evolved together. And let's see here. Documents, downloads. Okay. Here we go. Okay. Photosynthesis happens in the chloroplast. Here's a diagram of our chloroplast here. 3 membranes in the chloroplast, 2 on the outside, 1 on the inside, all folded up. The space in the innermost part of a chloroplast is called the lumen. The membrane, the third membrane that folds up in the chloroplast is called thylakoid. A stack of thylakoid membranes is called grana. And the space outside that stack is called the stroma. Stroma is where the Calvin cycle happens, and the electron transport chain that creates a proton gradient pumps protons into the lumen. So light is received by chlorophyll molecules. They get excited. They pass their excited energy onto an electron in the reaction center, which gets kicked out in a long electron transport chain, just like we saw in mitochondria. Those electrons are going to go this way across the membrane, and protons are going to go this way across the membrane. So electrons are losing energy as they get passed down the chain. That's used to move protons into the lumen. Okay, the electrons that leave this chlorophyll are replaced by splitting water, releasing oxygen as a byproduct. Electrons that leave photosystem 1 end up on NADP to make NADPH. So the terminal electron acceptor in photosynthesis is NADP to create NADPH. In respiration, in mitochondria, it's oxygen to make water. Chloroplasts have a F1, F0 ATPase just like mitochondria do, and they use it in the same way mitochondria do to make ATP. So you get ATP out of harvesting light, you get NADPH out of harvesting light. You can use those to fix carbon. And that's done in the Calvin cycle, which happens in the stroma. CO2 comes in through the stomata in the plant leaf as a gas from the atmosphere. It's added to a 5-carbon molecule called RUBP to create a 6-carbon molecule by an enzyme called Rubisco. Those 6-carbon molecules break down immediately into 3-carbon molecules. One of those 3-carbon molecules gets pulled away, and then you can put these 3-carbon molecules together to make things like sugar, starch, cellulose, build the body of the plant. The plant can store that energy for when there's no light. And then you regenerate that 5-carbon molecule and you go around again. Okay, so there's a very quick review. Hopefully that's helpful, and good luck on the final.